All right, great. Welcome back, everyone, to the afternoon session. We'll be starting off by discussing some of the challenges associated with data collection, aggregation, and sharing. The objectives of this particular session are to discuss how big data can be leveraged to identify patients, to enable patient stratification strategies, and also to step back and discuss some of the challenges in data collection and data sharing, especially from clinical trials with small patient samples or small patient numbers. And we have two really great speakers that will help us think through some of these issues. The first speaker on the docket is Dr. Larsson Omberg, who's a physicist by training, has spent time in computational biology, is currently vice president of systems biology at Sage Bionetworks, and has been involved with quite a few consortia related to data sharing and data analysis. So please help me welcome Dr. Omberg.
Thank you very much. It's a pleasure for me to be here. When I got this invitation, my first thought is, well, I don't really do regenerative medicine, and it's been over 5 years since I published a paper in the field of stem cell research. So at first I was at a loss, but then when hearing about systems approaches and the goal of this, I'm very, very honored to be here and speak to you today. And I'm really going to be talking about open models and alternative models for data sharing, and where there are not enough and when we need to do more, and some of the incentives that you need to build into that. And a little bit of background about me. So I work at an organization called Sage Bionetworks, and we're a nonprofit in Seattle, Washington. And really, we have a belief that modern science is a team sport. And by harnessing the power of open science, we help research communities develop reliable methods and outcomes to understand human health. And really it comes down to is that things have changed in the field of biomedical research. And what we've seen, and this is actually an old article from 2007, that we're seeing a dominance of team production of knowledge. And it's important to note that actually biology is actually kind of behind in this. This was something that was led out by some of the other hard sciences. I think physics and astronomy grew into large collaborative efforts early on because of the cost of science. And this is the same thing that we saw in the early 2000s up until today happening in biomedical research as well, is a lot of times research gets expensive. And obviously in genomics and in the omics fields, it's the data production. And it used to be that it was expensive because of the technologies used, and now it's expensive just because of the large sample sizes that you need to be able to have an effect. Interestingly, though, a couple of years ago, there was a paper that came out that looked at the impact of different types of research. And what they found is that large teams tend to develop and smaller teams tend to disrupt. Most of the work that we do at Sage Bionetworks is in the large team component, which is working in large consortia, working together and enabling them. But I might talk a little bit about this disruption as well, when we're having individuals working and coming up with new ideas. And I'll talk specifically about an example of that. Now, if you notice, I've used the term open science a lot. The reason I haven't used the term open data, because open data is just one component of many, and it's not enough to think about open data as, by itself. We have to consider it together with other aspects. Open data has grown significantly. NIH came out with an Omics data sharing policy that requires all data to be open after 6 months after generation. The idea of fairness, that is not just making it open, but making it accessible as well, has become a key driver of many research efforts. Out of curiosity, I did just a look at see how open data has evolved over time by looking at Google Trends a few months ago. And I noticed, it really started growing around 2010 and became more and more popular up until, and basically steady state from maybe about 2018 or something like that. One of the side effects of this, interestingly enough, though, is that a lot of this effort and searches are from the District of Columbia. The government really cares about open data. And this is something that, you know, we've observed as well. A lot of scientists are reluctant to to work in open methods. And a lot of times it comes down to, they feel it as a threat. And so, you know, obviously we want things to be open, but then scientists might sometimes are afraid of it. I think that's changing a little bit, but a lot of it comes down to incentives as well. What is my incentive for collaborating and sharing my data? And interestingly enough, we have examples of, you know, people who've been very anti-open and then realizing, oh, wait, if I am forced to be open, it also forces everybody else to be open. And now all of a sudden I can have multiplicative effects in this. And we're seeing this a lot among younger researchers, particularly. Now, it's important to note that open is not enough. And this comes back to the incentive problem as well. So in physics, they discovered this by making large numbers of data sets available from astronomy. A lot of that data went fallow. People weren't using it. You need other structures to actually make people work on the data as well, because data in itself is not enough. You want the incentives and the insights from the insights from that. It is worth noting, though, it's a good first step, and oftentimes is a required first step. I will talk a little bit at the end about a couple of models where open doesn't work, and you know, the introduction talking about the fact that, you know, when you have small trials, those are situations where open might not work because there might be sensitivity to sharing those data. So I'll talk a little bit alternative models at the end of this talk. So let's get concrete. At Sage, we work a lot in large collaborative projects, oftentimes consortia. We primarily work in the fields of cancer research, neurodegeneration, as well as neuropsychiatric diseases, but we do some other smattering of work as well. So I'm going to walk you through a specific example and then a specific example with that, and then I'll shift gears and talk about collaborative challenges as well. So my first example is going to be the Accelerated Medicine Partnership in AD. So this is a public-private consortium that was formed 7 years ago, I believe, looking at the problem in Alzheimer's disease of lack of targets. Alzheimer's disease, obviously, as most of you are aware, is a devastating disease. We've cured it 100-fold over in mice, but never in humans. And every single clinical trial has failed. And even though billions of dollars have been poured in it, both by pharma as well as by the government. So one of the suspicions is that pretty much there's a monoculture. A lot of effort has been focused on a few set of targets. And so the AMP-AD was initially formed with the idea of expanding these targets by looking at systems biology approaches and identifying new targets. And that's since grown quite a bit, and we now act as a coordinating center for multiple of AD-related, from Model AD, which is developing mice models, Resilience AD, looking at resilience, Move AD, which is looking at vascularization, as well as Psych AD, which is looking at the interaction between psychiatric diseases and Alzheimer's disease, as well as Treat AD, which is taking these targets that have been identified and trying to develop target-enabling packages to help with the preclinical development of drugs. But of course, all of these start with data, and we currently maintain a data repository where we're over 15 different omic data types, over 7,000 donors, and really it boils down to gathering data from multiple different sources together. But really, if you look at it in terms of, it's a pyramid, right? The data is the first step, and all of this data is open access, so anyone can come and use it. But then from that, you build analytical tools that are run on top of this data. You get analytical results, but then in the end, you get targets or insights, which in this case, in the AMP-AD, we're looking for targets. Now, I'm going to talk about a specific example within this, which is basically an effort and kind of a model for how we work together, which is looking at an RNA-seq analysis that was done within the AMP-AD consortium. So the AMP-AD was formed with 6 different academic groups. Now it's significantly larger than that, but there are multiple groups that were generating the same kinds of data. So RNA-seq on postmortem brain samples. And Problem was that this data was very, very heterogeneous. It was difficult to use because each of the groups had collected using different technologies, different analytical approaches, and so it wasn't directly comparable. And so a large effort was done to homogenize this by having several of the groups working together to create a canonical data set that then can be used in downstream analysis for insights. And this is one of these examples is that of the 6 groups that have submitted to be funded under the AMP-AD umbrella, 5 of them had proposed to do the same kinds of analysis, but they were all going to do the same kinds of analysis with their own data set. But by pooling this data set, each of them could now do their own analysis on at least the same data set. And so we formed a working group that basically would take each of those methods, they could each do them themselves, and then basically to start doing consensus modeling across this. And specifically, this is on a co-expression network analysis, as well as differential expression analysis. So what we ended up doing was having each of the teams running their own methods, as well as some of the pre-published methodologies that are out there, and then doing a consensus by comparing all of them and coming up with a common set of output, so that it'd become easier for people to know what is the canonical set of networks that exist, and then also to focus on what are the differences. So like comparative module analysis, seeing those that are different between each and the other. And then combining these two, we ended up seeing that there was actually a large amount of overlap between each of the groups. And this is looking at the consensus networks, and those consensus networks corresponded to known AD biology. So for example, cell-specific AD biology, as well as general AD biology. But interestingly enough, there's a large subset of these consensus networks. So these are things that we find commonly across all the different methodologies, or strongly across most of the methodologies, but did not correspond to previous known AD biology. And so this is kind of highlighting that the dark matter that might exist within AD biology. And, clearly of interest if you're looking at new possible targets or identification of source of disease. Of course, taking this and having this be useful takes another large amount of research, however. So identifying new targets, combining this as well as you want to be able to eventually get to developing targets that could be used in drug development. So this is where NIA has invested in the Treat AD effort, which is the idea is basically to use the targets that were identified in the AMP-AD consortium, identify which ones might be druggable, and then develop tools so that the people who are doing drug development can take it to the next step. And so currently that means taking the targets, maybe sometimes expanding those into a larger set of possible targets based on druggability. So going back to the modules that appear, and then identifying the best targets within modules. I'm going to shift gears a little bit here and talk about another type of collaborations that we work. So what I talk about there was, the idea of having working groups working together, and that's Dream Challenges, which is a community challenge and biomedical benchmarking. One of the things that happen a lot when someone develops a new method is that they develop the method, they validate the method, and they test the method all by themselves independently. And what ends up happening when they publish it, everybody seems to be doing better than average. Or as Garrison Keillor used to say, where the women are strong, all men are good looking, and all children are above average. We can't all be above average. So the idea behind Dream Challenges is to separate the idea of development and benchmarking. But it also has some other benefits as well, which is basically, it avoids the self-assessment trap that I talked about, but it also creates incentive models for people to work together. And by posing it as an open problem, you can quickly explore a very large space of solutions, i.e. you're crowdsourcing solutions to problems. And so I'll talk about a specific example, and this is not out of genomics, but actually out of digital health. So I've been running a study, we launched it in 2015, of recruiting people with Parkinson's disease and trying to measure their symptoms using a smart device. So using the accelerometers within a smartphone, as well as interactions with the screen to measure different aspects of the disease. And being from an organization that cares about open, we actually released this data, the first 6 months of data, months after those 6 months were up, before we had any chance to analyze the data ourselves. And we made it a public resource. And we had, within about a year, we had a little over 130 individuals who requested access to the data from 35 different institutions. And at this point, there's over a dozen publications that have come out. Without naming any particular publication, I'll mention several of these publications are quite wrong about their insights on this data. And I would partly blame that from the fact that a lot of the people who came to this were very good at machine learning, but not necessarily well versed in the disease. And they did not consider the covariates that might affect the disease, as well as the confounders in the data. And so what we decided is to turn this into a challenge where we could benchmark different methods, where we would control for the things that we knew were issues after we even analyzed the data ourselves. And so we posed it as a question: can you build a diagnostic digital biomarker that uses the accelerometer data to tell whether someone has disease and also what the severity of their disease is? And once doing this, we ended up having another 350 individuals accessing the data and building models on top of it. And what we did is, as part of this, we actually convened a group of people, experts that we knew in the field. And this included people from Verily, Harvard Medical School, University of Luxembourg, University of Rochester, nonprofit working in accelerometer and digital wearable data. So basically experts in this field. And we all did our best to analyze the data. We had, of course, spent years with this data at this point. And what we found is that as a group, we were doing about a diagnostic biomarker with an AUC. So basically the accuracy, how well it could do, was about 0.69, which is not very good at all. But as soon as we post this out, where 350 people had access to the data, and they're all building their own models, they were able to build measures that had an AUC of 0.84. So within weeks, we were beat as a group of experts. And by the end of 3 months, we had over a 20% increase in the performance. And so this is one way where the incentive of competing actually works to make people to do better. And then, of course, the important part is making this open as well. So everybody's algorithms were needed to be open source, and we then turned those into Docker containers so that people could reuse these in their own analysis. And this is, the other component of the part. It's not data alone, but data and methods that need to be open. Now, unfortunately, a lot of the data that we need to answer impactful questions are not open data. There's restrictions such as HIPAA in the US, and the GDPR has put restrictions on how we can access and use data within Europe, as well as their ethical implications. You need to consider, how was this data consented, and are there ways we can have data that can't be fully open be used? And this comes down to data governance, which is the freedoms and constraints and incentives that determines how parties can work to manage themselves to work on ingress, storage, analysis of data, tools, and methods and knowledge. And really, this comes down to, there's communication, negotiation, and terms that between groups, but it also involves softwares and technologies. And there's actually several different possibilities for this. So this is a figure out of a white paper that we've been working on, which looks at different models of people working together and seeing how they relate to what kind of analysis you can do and what the freedoms are with it versus how easy it is to access. And, obviously at the bottom, closed, restricted data being data that only a very small group of people can work on, to the most open, which might actually involve citizen scientists, or open source data, which is what mostly I've been talking about here, where it's data that's open. There might be some restrictions on the data, but anyone can access the data anyways. So I'm going to talk about one of these models, which is the idea of models to data, and I'm talking about it because it fits into the premise of challenges. And the idea here is that if you have a very specific problem that you're trying to solve, instead of having people access the data, you can have them submit their analysis and have the analysis done. It doesn't work for general purpose analysis, but it works for those where you have specific questions that you want to answer. And one of these was one that we ran a few years ago, which was a digital mammography challenge, where we were looking for people to diagnose whether someone was going to develop cancer within 1 year. And part of the restriction is this is PHI data. A lot of these things have scribbled notes on them with names on them. And on top of that, it's images from a clinical center. We had over 140,000 images from Kaiser Permanente. And then on top of that, it's also very large data. It's 14 terabytes. And if you want something to be accessible, you have to democratize the compute and allow even those individuals who don't necessarily have a cluster available to them to do this kind of analysis. And so what we have developed is this idea of having a model to data where people are submitting their models. We execute them on their behalf. And in this challenge, this is actually one of many that we run. We had a dual cloud. We used IBM and Amazon. And people used GPU machines. And then in the end, you end up with something that is not only the model that people have trained, you actually get something, a Docker container, which is something that can be reused or at least examined by other people. I'm running a little late, so I'll stop here and say, just in conclusion, open is not always enough, but with the right incentives and structure, we can do better science together. Thank you very much.
Thank you, Larsson. That was a really, great sort of framework and proposal for potentially moving forward to enable that type of data sharing. Really helpful.